Next couple of minutes. Just give folks like a couple more minutes to join the call. To start us off, now that we've hit the 50 participant mark. So, hello everyone, and welcome to UGA's Institute of Bioinformatics seminar series. Today, I have the honor of introducing Dr. Michael Landis from Washington University in St. Louis. Before this, he was a Yale Donnelly postdoctoral fellow and a recipient of the Ernst Mayr Award. Dr. Landis's research focuses on understanding how biodiversity changes over time. His lab develops probabilistic models for evolution and contributes to open source software that investigates questions around historical biogeography, ecological interactions, and pathogen spread. Please join me in welcoming Dr. Landis to UGA by either using the clap reaction in Zoom or turning on your video for a couple of seconds just to wave hello before you get started.
Yeah, we'll do a truly manual clap rather than the
Yeah.
so thank you. Thank you, Guppy, for the introduction and for the invitation as well. I am not good at PowerPoint. I'll see if I can share my slides properly. One second. I think this will do it. How does that appear? Is that okay? Great. Okay. Right. So like Guppy said, my name is Michael Landis. I'm here at WashU in St. Louis, and my background is in statistical phylogenetics. Um, Now, I'll be talking a little bit about phylogenetic models. Phylogenetic models are useful for all sorts of things. Models in general are valuable for the ability to formalize conceptual models that we might have as biologists, for simulating data sets, testing hypotheses, estimating model parameters, finding the best model, and with phylogenetics, reconstructing the past, like species relationships, ages of lineages, ancestral states, things like that. The types of phylogenetic models that I'll focus on today are diversification models. With diversification models, we're describing a stochastic branching process. And in particular, it's a branching process that also has states associated with it. So we're going to have different, say, branching rates or speciation rates that depend on state, different extinction rates that depend on state, and so forth. So here we have one realization under a branching process, under a diversification model with 4 steps. By the way, what can you see my mouse when I wave it around, or is that invisible? Okay. So here we have one phylogeny, a realization under the model, and lineages might change through states, the blue dots, and you can see it goes from, say, a black state to a green state. Lineages can go extinct. They can be sampled, which is important for virus phylogenetics, and so on and so forth. And with these models, the idea is that you could potentially ask important questions that relate to the states and different rate estimates. And if we set up our model properly and we estimate these rates, we can talk about equalities or inequalities among rates and test those hypotheses quantitatively. As Guppy mentioned, my background is in phylogenetics, and I mostly actually work on the macroevolutionary time scale, and I'm interested in things like how do plants adapt to different temperature regimes, or where did species live in the past at the continental scale. But what I'll be talking about today is mostly focused on epidemiology for various reasons. Now, I mentioned that phylogenetic models are useful for many things, but the applications for these models often historically, they've relied on having a likelihood function. Now, normally when I present this material, it's to biologists, but this is a bioinformatics department. So I'm going to abbreviate some of the ideas that concern likelihood. I'm just going to give a refresher for those who need it. And otherwise, I'll spend my time talking about phylogenetic modeling and alternative inference methods. So, statisticians love things like dice and coins and urns. So let's take a simple model where we're simply flipping a coin 3 times. And we want to ask, what's the probability of observing 2 heads out of 3 coin flips? Now, one thing I could do is if I have very few coin flips is I could simply enumerate all the outcomes and count up the number of times I get 2 heads, right? That's 3 out of 8. Not too hard. In general, this problem can be reframed as a binomial sampling probability. And in this case, I need to think about all the different ways I could order coin flips you know, um, uh, for the different three coins I've flipped and the possible outcomes. And then I need to think about the probability of having a head or tail for those different coin flips. So here, what I might be interested in hypothetically is, can I estimate the fairness of the coin given some number of coin flips? Now, once I start getting into larger possible outcomes. So the binomial, that binomial distribution's probability is really critical for generalizing our ability to fit models to the data sets we actually have in our hand. So here's a simple plot of a coin flipping experiment where I get 61 heads out of 97 flips. And if I asked what's the probability of it being a fair coin given those outcomes, I would say it's a relatively low probability or likelihood for that observation, whereas the maximum likelihood would want me to have a fairness of 61 divided by, that's a typo, 61 divided by 97, not 87. Okay. So we can use this to fit models and learn parameters from data in principle. Now, with modeling, my perspective and many people's perspective is that we only know the truth through simulation. So let's say I perform many, many coin flipping experiments, and here's a histogram of outcomes for numbers of heads after flipping 90, sets of 97 coins. If I have a probabilistic model, I might want to ask or estimate what is the fairness of the coin across all of these experiments. If I know the right model, I have a very good chance of actually fitting my likelihood model to the data generating model. And I can do this with simulation because I know what the simulating model is and I know what the inference model is, and they match perfectly. In practice, for empirical scenarios, we basically never expect to have a likelihood model that perfectly matches the data generating model. This is the all models are wrong, but some is useful perspective of George Box, right? So we expect that when we actually go to analyze data sets, we will have a misfitted model, a misspecified model with respect to the data generating distribution. Okay. Now, I've been talking about coin flipping. This entire analogy applies to phylogenetics as well. For example, suppose I wanted to estimate a parameter that describes the expected numbers of mutations along branches. Same procedure, generally speaking. I have a data set. I want to basically fit my distribution to match my observables. Okay. Now, it turns out that in phylogenetics, we have a problem because from the get-go, we don't have the equivalent of a very simple binomial distribution like we had for the coin flipping example. For phylogenetics, if you take it naively, we want to basically marginalize or integrate over all the possible histories that could be in our data set. So for example, if I have 5 taxa, 1, 2, 3, 4, 5, and they have a single nucleotide with different states for each taxon or species or gene copy, and those nucleotide states are A, A, C, A, T, in reality, I don't know what any of the ancestral states are for nodes 6, 7, 8, or 0. So I need to average over all that uncertainty in the past. The way you would do that typically is just by summing over all of the possible values for variables that you want to integrate out. And that doesn't scale up very well when you have large trees. Now, in order to make this type of model tractable for phylogenetics, somebody clever, and in our case, we're fortunate to have a person such as Joe Felsenstein, develop algorithms that basically let us integrate out the variables that we can't observe. Now, I just want to emphasize this is a really fundamental problem in phylogenetics because there's lots of models that we can think about as biologists, but not all of those models are guaranteed to have tricks like this one that allow us to actually extract tractable likelihoods from those models. Okay. Now, I'm going to step back and recenter on the actual biological problem that will be the bulk of the rest of the talk, which is viral phylogeography. So one thing we can do is we can go and extract sequences from lots of different infected individuals for the viruses they're infected with, reconstruct their phylogenies. We'll know something about the time those viruses were sampled. We'll know something about the location where those viruses were sampled. And in principle, we should be able to reconstruct something about the past of an outbreak, right? For example, if we knew many viruses were in North America and they're closely related genetically, we might reasonably expect that their most recent common ancestor is also from North America, um, barring any other types of evidence. So what we'll do is like I said, we'll collect lots of viral sequences, estimate a phylogeny, associate times and locations with those sequences, and try to reconstruct the past, allowing us to ask questions such as, do viruses tend to move from one region to another region or vice versa at a higher rate, right? Do they replicate faster in one region versus another region, and so forth? Um, when we start thinking about how outbreaks occur, most outbreaks in human populations are actually caused by spillover from an animal population into a human population. So there's also underneath all of this, there's a fundamental ecological context to why do certain outbreaks happen in certain places and how do they spread? It really depends on not just what humans live there, but what other species live there, right? That complicates things a little bit. Because in order for spillover to happen, you have to have some amount of sympatry, biogeographical overlap between species, potentially overlap in terms of their ecological niche that actually creates the opportunities for spillover to happen. So now it's becoming a little bit more complicated compared to our Felsenstein pruning algorithm, where all we care about is AACGT and so on, right? There are multiple layers to what might impact spillover. So we've talked about ecology and where species live. It also depends on the sort of the cellular level defenses of the host. It depends on life history traits and how population dynamics play out in different species, and so on and so forth. All of these factors matter, right? And with modeling, we're constantly balancing how much detail do we need to answer the biological questions. But with modeling, there's a separate problem, which is how do we have the simplest model that is tractable that gets at those questions as well? And this is a real barrier in designing models, is this computational tractability. So let's say you have a beautiful model in mind. You've spoken to all of the biological experts and they agree it needs those features. If I were to rank, and this is difficult to rank in any particular order, but if I were to rank from easiest to hardest tasks, I would say that defining models is often one of the easiest parts of model design if you ignore the other downstream parts of it, such as how difficult is it to write software that will simulate data or realizations under that model? Or how difficult might it be to get a likelihood function out of a relatively simple model versus a very complex model, such as the one I just showed, where you might have ecological factors, cellular factors, population dynamic factors, and so on, that influence the spread of viruses in space over time. It gets complicated, right? And it turns out that many of the phylogenetic models that we actually apply uh, are ones that have likelihood functions, which means they're tractable likelihood functions, which almost necessarily means they're extremely simplistic representations of biology. But when you talk to biologists about what types of models they wish our models could have, they will list out types of properties that induce things such as large or unusual state spaces. This will break tractability. Complex types of rate variation. This typically breaks tractability. Trees within trees, such as multi-species coalescent models and so forth, or gene family evolution. This will break tractability, and so on and so forth. So we have a fundamental trade-off, in my opinion, where on one hand, we might seek out biologically realistic models, but on the other hand, we face reality that we can't always get the mathematics or the computation to yield these likelihood functions that we want for inference. That's a real fundamental trade-off we face in modeling. Not just phylogenetic modeling, but modeling in general, I'd say. Now, during the, yeah, at some point during the pandemic, a few years ago, my kids had gotten up early and life was complicated and they were desperately wanting waffles one morning. And I went to the cupboard and I went to the cupboard to look for batter mix and I had nothing where there's no syrup really. And I had to improvise something because otherwise there'd be a revolt in the household. And what I did is I basically fetched a piece of bread, put it into the griddle, pressed it, and poured honey on it and guess what? The children were perfectly happy. And I'm not sure, I think I'm probably reinventing history a little bit here, but I had a realization that what I'm interested in as a modeler in terms of certain theoretical properties or clean solutions that rely on the math is not necessarily what people want in terms of users, the biologists who actually want to apply these models to data sets. And the analogy in my mind is that, you know, what most biologists want is a parameter estimate. They want to be able to test a hypothesis, such as the ones I talked about earlier, right? They don't necessarily want an elegant algorithm that lets you integrate over ancestral possible states in short amounts of time. They want the parameter estimate that that algorithm lets you get at. So for me, there's a connection between this notion of what can we get expeditiously and what do we actually need to solve the problems that we're facing today. And I think that's especially poignant in terms of epidemiological problems. We want to be able to get estimates for rates of spread today, yesterday. We want to know how people are going to be harmed by these outbreaks, right? So we can't always wait for, say, 6 months for a Bayesian MCMC analysis to run to figure out how dangerous the pandemic was, right? We need results faster in some cases. So I started working with a person I'll introduce in a moment here, Ammon Thompson, on ways of using these deep learning methods that have become more and more popular over the past few years to aid likelihood-free inference under phylogenetic models. And with viral phylogeographic models, we certainly have some of these complex features that I just described, such as complex types of rate variation, which hosts are available, which are susceptible, which are infectious, and so forth, and how do they interact in space over time. How does extinction, such as the recovery of hosts, or the location in which those viruses are spreading, influence branching processes, and so forth. So the bulk of the work that I'll be talking about today is related to this paper, which was published last year. And what we were seeking to do is coming up with not only some inference strategies for how to do likelihood-free inference with deep learning, but also to compare the quality of the inferences we get with deep learning against the gold standard inferences we can get through Bayesian inference. And I'll take a second to comment that my background is in Bayesian phylogenetics. If anything, I am biased towards Bayesian phylogenetics. And so when I started on this project, my expectation was that we would find ways to break deep learning. So as I'm going through these results, I just want you to keep that in mind. I am definitely a skeptic of deep learning methods, and I was trying to find ways that would potentially show cases where deep learning fails, but classical likelihood-based inferences succeed. Okay? Now, the three main questions we sought to ask were, how can deep learning methods be used to estimate phylogenetic model parameters? How can simulations be used to train neural networks? And how will neural networks behave when they're trained on the wrong model, when we introduce model misspecification? Because that's going to emulate what we actually face when we're doing empirical data analysis, right? We don't know the true model that generates epidemiology, epidemics, right? We only have our simple mathematical models. So I'm just going to skim over this part, but many of the questions that are interesting to epidemiologists are fairly simple and relevant to public health policy, such as where do outbreaks tend to start and when do they tend to begin? How do they start, under what conditions, such as from which species do they spill over, and how quickly do these outbreaks spread among populations? Now, when we're actually trying to when we're trying to answer these questions, we have very sparse sampling. We don't get to sample every single virus that infected every single host over time. We often have a subsample of these infections, and we want to reconstruct the past from those infections we do sequence to try to generate a historical picture of the process. So here's an example where we might have, it looks like 6 different samples from different geographical locations. We can sequence them, identify their locations, and then build phylogenies and reconstruct movement of viruses. These types of models are applied all of the time in the phylogenetics and epidemiology literature. Here are three examples using epidemiological models to look at the the rate of importation of viruses into the UK from different European nations for COVID. Here's one looking at the timing of introductions of different Ebola outbreaks into different countries. And here's one looking at how HIV spread throughout an African nation in the 40s and 50s. One thing to note is that for some of these epidemiological applications in phylogenetics, they rely on what's known as the SIR model. The SIR model, which stands for susceptible infectious recovered populations or compartments of populations, are basically ways of tracking how pathogens spread, infect, and allow, either kill, that's a form of recovery, or allow hosts to recover and survive from infection. And one thing that most people who have heard of SIR before will be familiar with is the prevalence curve, which is basically a way of monitoring how many infections are in a population at a given time. So if you remember the battle cry, flatten the curve from the early days of the pandemic, the idea was that if we can reduce the height of the prevalence curve, we can basically make sure that our hospitals aren't saturated with infections and everybody gets the care they need when it's evenly distributed. Now, one thing to know about SIR models is that in the earliest phases of an outbreak, which I've colored pink here for nice, during that phase of outbreak, we have exponential growth of infections. There's basically nothing stopping viruses from finding new susceptible hosts to infect. And because we're in this regime of exponential growth, phylogenetic SIR models are actually identical to birth death models. When we get into the later phases of an SIR model, when we start to saturate the number of infections and there are fewer and fewer susceptibles to infect, we see the prevalence rate declines. And in fact, this is going to induce a reduced per capita infection rate over time. And because of this, our standard birth death models are somewhat difficult to fit to SIR dynamics over the entire timescale of an outbreak. There are lots of very sophisticated and complicated ways that people get around this problem. But in terms of simple likelihood functions, we do not have one for this later regime of a pandemic. We only have tractable likelihood functions for this first part. Okay. Now, this is one of the simplest, if not the simplest SIR model you can imagine. There are hundreds and thousands of different papers out there describing more complex SIR models with more compartments. So here's one which is looking at differences in vaccinated versus unvaccinated individuals and what type of hospitalization they are given access to, and how does that affect recovery versus mortality. And individuals can lose immunity over time, and so on and so forth. If we can't get likelihood functions for an SIR model under this part of a pandemic or an outbreak, we certainly won't be able to do it for this type of dynamic, right? So there's some gap between the types of likelihood models we actually have, the types of models we have likelihood functions for, and the types of models that epidemiologists might want to apply to real genetic data sets. There's a gap. So we began looking into different likelihood-free methods for trying to bridge this gap. For various reasons at the time, we were really drawn towards the newer deep learning methods that had become popular around 2020, 2019. And some reasons for this that we're drawn to these were that they appear to be very generalizable. There are other methods that you can use for likelihood-free inference, such as ABC methods or in-law methods, and so on and so forth. But one nice feature of deep learning methods is that, one, they have a certain promise of being generalizable, and two, they don't make strong assumptions about the mismatch between your data generating distribution and your inference distribution. So we thought this might be nicely applicable to a broadly underexplored class of models, such as these phylogenetic epidemiology models. And many people have been publishing in this area relatively recently from my neck of the woods in statistical phylogenetics. We're just in the early steps of trying to figure out what these methods are capable of, and it's still relatively poorly understood. So like I mentioned earlier, my background is in Bayesian phylogenetics. And I and others have been seeing these papers coming out, and they seemed too good to be true. And so me and my collaborators, we wanted to assess in as fair a manner as possible, how well do they perform under different realistic conditions. And we thought the best way to do that would be to compare gold standard methods such as Bayesian methods to deep learning methods to understand, to identify when and where we see gaps in performance and potentially diagnose for what purposes are deep learning methods actually useful. Now, I'll give a few slides here on the basics of deep learning, but I won't get into too much depth. One way is essentially what we're trying to do is we're trying to construct a function that allows us to take a phylogenetic data set as input and use it to predict something such as a set of evolutionary rates as output. And one nice thing about deep learning methods is they are capable of making predictions using nonlinear functional relationships. So one way I often think about deep learning methods as highly nonlinear regressions of regressions, highly nested sets of regressions of regressions. And in principle, what we'd like to be able to do is take different phylogenetic data sets, which we've colored here as points, think of them in some type of continuous space, and use them to predict the y-axis, the evolutionary rates or other things we're interested in. I'm just going to get, for people who haven't worked with deep learning before, I like to show a quick animation to give people a sense of how does this work. This is a really wonderful website where you can play around. It's called playground.tensorflow.org. So you can certainly play around with neural network architectures and training settings and so forth. And here what we're going to do is we're going to try to train a neural network to discriminate the orange dots from the blue dots. Okay. And I've clicked the play button here, and we're going to see the number of training epochs. This is the network trying to optimize a very large number of internal parameters that basically form associations between different nodes in the neural network by controlling the weight of importance and the biases of signal across adjacent layers. And what we'll see is that the colors and the weights of these links, which are the edges, will shift over time. And as the neural network begins to train, it will eventually find optimal patterns that are best at discriminating the orange from the blue dots. And the optimization criterion it uses to discriminate is this loss function, which we see up in the upper right. As training proceeds, we reduce the loss score, and the loss score is basically a metric that where we know what the true answer should be, and we know what the network is predicting, and we can try to minimize how different those predictions are from the truth. So when our network is performing well, and it's been trained to perform accurately, we are minimizing the number of times we get the answer wrong based on the true examples. So in general, what we need to train a neural network is we need for supervised learning, which is the technique we'll be using. We need a data set that has training data and labels. So we need inputs and outputs, which will allow us to form correlations between, so the neural network makes accurate predictions. The neural network is an architecture that describes how different optimizable variables relate to each other. And for the most part, this is what people refer to when they say neural networks are a black box or deep learning is a black box. We have an extremely over-parameterized model, and we're trying to find good parameter values that lead to accurate predictions. And finally, when I say accurate predictions, we're trying to train the network so that when it predicts a value from inputs, it's basically minimizing the error between what we know is true and what the network thinks the value should be. Okay. Now, for phylogenetics, we have at least three problems here. And as I see it, the three problems are, in general, we don't have very good ways of estimating, measuring similarity between phylogenies. So if I want to talk about the difference in a one color versus a different color, I can convert that to an RGB scale and talk about some distance, say, in Euclidean space. But a phylogeny is a very complicated mathematical object. It's a combination of discrete and continuous variables. So we need some way of getting that into a distance-based space so that we can apply distance-based metrics to it. Related to that is it's not, there are different ways you might actually take that phylogenetic object and put it into this regularized space. Neural networks typically need inputs to be in what's called a tensor format, which you can think of as a high dimensional array with regular numbers of elements per dimension. So it's a block matrix or an n-dimensional array. And then finally, we need to have some way of generating true phylogenies for training, because the entire reason we want to do phylogenetic inference in the first place is because we don't know the true history of the outbreak. So we can't go out into nature and find millions of examples of true outbreaks. We somehow have to generate sets of hypotheses for what those outbreaks could be to train our network. So these, in my opinion, are the three main challenges we face. One thing about phylogenies is that tree space is vast. So when you start getting into, let's say, 20 trees with just 20 taxa, 20 tips at them, at the ends. I chose a number where I actually don't know what 1, 2, 3, 4, 5. Oops, 5. 1, 2, 3, 4, 5, 6, 7. So what's that? 10 to the 8 to the 10 to the 21st power. 8 times 10 to the 21st power in terms of numbers of possible topologies. And what I want to emphasize here is that once you start getting to reasonably sized trees, such as 20 species or more, or 20 virus samples or more, you're almost guaranteed to have a unique type topology where you would not expect another tree with 20 taxa to have the exact same configuration. At the same time, trees are inherently recursive objects, which means that you should be able to pull out these repetitive patterns within trees, which can help you form bases of comparisons between them. Now, I'm bringing this up because this issue of how do you compare trees and the uniqueness of each tree is related in many ways to image processing. So in deep learning, many people use convolutional neural networks to extract local features from images, a 2D matrix, in this case, a 2D grayscale matrix, and turn them into very low-level features such as vertical versus diagonal versus horizontal lines. And then you can turn those features into meta features, which start to describe eyes, ears, noses, and so forth. And then construct meta features of meta features, which starts to give you things that resemble chins and foreheads and faces and so on. And so similarly, we should be able to apply CNNs to extract not just local features of phylogenies, the equivalent of a line, but we should be able to aggregate that information to generate meta features such as subclades and larger subclades until we start to recompose the entire tree. So we want to develop an approach or use approaches that let us extract these local features so that we can aggregate signal across our large numbers of possible trees we might encounter. The other problem we face, as I said, is we need to somehow get our trees into a space that makes them easy to compare. And the way that sorry, I went too quickly. Again, with neural networks, you generally need to input your information to the neural network in some type of matrix-like or tensor format. Now, there are various ways that you can encode phylogenies into matrix formats, such as a patristic distance matrix. But there's a lot of redundant information in that type of matrix. This nice paper by Jakub Wasnaka and others in 2022 introduced a vector encoding where you take a tree with branch lengths measured in time. And you can perform a series of transformations to basically encode it into a vector. And this vector encoding will be equivalent to n times 2 the number of taxa, the number of tips in the tree, which makes it relatively compact. It's effectively linear with the number of taxa in terms of storage. It's bijective with respect to the original tree, meaning it perfectly captures all information that was encoded into the original tree. And if I give you the tree and encode it, then I can take the encoding and regenerate that tree I had in the first place. And then finally, it's vectorized, which means we can actually put it into a tensor format, which makes it suitable for training neural networks. With Ammon Thompson, my collaborator on this project, we extended this format to include states for the phylogenetic tree. So we don't only include branch length information and topological information, but we also start to include locational information, such as where did we get these different virus samples. Once we have a format like this, we can begin applying our CNN approaches, which will basically scan our tensor to look for local patterns, local features that can be used to reconstitute information as points in a high dimensional space. And we can apply all sorts of techniques such as standard PCA approaches to get a sense of what does tree space look like in this I think in this case it's a 1000 dimensional space. And these blue points represent individual tree samples that fall within that PCA space. Okay. The last of the three problems I mentioned is that we don't have examples of the truth in phylogenetics in general. And this is a real problem. Again, we want to do phylogenetic inference because we don't know the truth. Now, with likelihood methods in general, we are assuming a model that describes how things evolved. So what approaches like ABC and other likelihood-based deep learning techniques use is they take the model, generate realizations under that model, and then use the data sets you simulate from that model to form predictions about how should our data generating parameters here on the left correlate with different types of data patterns we can observe on the right. In a real empirical setting, the only thing we'll ever have is an input. We'll have a real data set. And what we'd like to do is to be able to take a real empirical data set and use it to predict what these data generating parameters are. So our goal here is to use our model to simulate data sets, which will act as our training data set. And then ideally, we should be able to form that correlation that I described earlier between data sets on the x-axis and model parameters on the y-axis. And we'll train our network to form that prediction if all goes well, right? Some people who are interested in deep learning might be curious about our network architecture. In my opinion, it's nothing crazy. We're not developing any novel architectures. We're basically using PyTorch, which provides building blocks for composing networks, and otherwise following best practices for the training procedure, such as having a relatively large training data set, making sure we're shuffling and splitting to produce test examples and validation examples to verify training worked properly. We also estimate what are called conformalized prediction intervals, and so we have a small calibration data set that we withhold. It looks like this should be 20%. I didn't add properly. For the network, we use ReLU units, mean squared error for loss. We use a pinball loss function for our prediction intervals. And then for training, we use an atom optimizer, mini batch with stochastic gradient descent, et cetera, et cetera. None of this is too complicated in terms of how complicated network architectures can get. What's actually interesting scientifically, I think, is the experiment that we run. And so we are going to run an experiment where we first train our neural network using a very simple SIR model. Okay. And this SIR model, we're training it only during the exponential growth phase of an outbreak. So under this condition, we do have a likelihood function that matches those modeling conditions as well. That's a birth death model. So we also have a Bayesian model that will have identical, that should in principle have identical inference properties as the neural network, assuming each behaves as advertised, right? And what we can do is now that we have two separate inference models, we have two separate inference methods, is what I meant to say, and they are both targeting the exact same model. We can then ask how do each method perform when we are applying them to when we apply them to data sets whose model assumptions match the inference method's model assumptions. So when the model is right. We can then also ask how do our methods behave when we apply them to data sets that were simulated under slightly different model assumptions, when we violate model assumptions, when we introduce model misspecification. Do they misbehave in the same way or do they misbehave differently? Now, I expect that when the model is right, when we provide both of them with a very simple SIR model during the exponential growth phase, um, Bayesian methods should basically always perform equal to or better than the CNN method. And I don't think it would necessarily be drastic overperformance, or outperformance, I should say, but I would expect on average the Bayesian method should behave better than the CNN method because the CNN method will be more vulnerable to not having perfectly accurate predictions because it's essentially an approximation of our likelihood function. When I give up both these methods the wrong data set, I would expect that they both will perform poorly, but I would expect more catastrophic failures for the CNN. And this is because the CNN is trained on a set of examples that it has seen before. Whereas the Bayesian model has a mathematical representation of possible data patterns. And those are hard rules that extend even outside the types of data sets that the neural network would have seen during training. So the Bayesian approach, because as a likelihood function, has a broader view of possible evolutionary outcomes than the CNN does, because it's a generalizable way of assessing probabilities of outcomes, the likelihood function is. Now, for our comparison, we're going to be using RevBayes with a very fast plugin for computing birth-death model likelihoods called TensorPhylo. I've been helping develop RevBayes for the past 15 years. So I know how to use this software very well. And I have the highest level of trust in terms of its correctness. So again, when we are looking at these experiments, we can essentially factor out user error on the Bayesian side. I really think we're running these analyses correctly. But you'll have to trust me on that. Now, I'm going to show you about 7 or 8 different figures that have elements that look something like this, okay? They'll be color-coded so that the neural network approach is in blue. On the x-axis, we're going to have a prediction for the neural network, and on the y-axis, we're going to have the true data generating parameter. If the neural network does well, we want the prediction to fall on the 1-to-1 line, okay? We'll do something similar for the Bayesian analysis. On the x-axis, we have the mean posterior estimate. And on the y-axis, we have the true value. And again, we want them to fall on the line. And then finally, we have a gold pattern here. And in this case, we're going to compare the estimates against each other. If the neural network and the Bayesian approach both agree, we also expect them to fall on the line, right? And for fun, we've also thrown in an ancestral state estimate for the location where an outbreak occurred. So in a 5-region system, for which of the 5 regions has the correct outbreak inference? So here's our simple case. We expect that our data generating model is a perfect match for our inference model. There's no model misspecification. So we really think that we can get the answer correct. both for the Bayesian inference and hopefully for the neural network approach. Simple model. So like I said, I'm going to show you a handful of plots that look like this. Here's how they'll be organized. These are the three columns correspond to different model parameters that are relevant for the outbreak. And it's the basic reproduction number, the sampling rate, and the migration rate between locations. The first row is the CNN estimates, the second row is the Bayesian estimates, and the third row is the degree of agreement between the two methods. And then down here is our spillover location. And notice that they generally look the same for the blues and the reds across panels and across rows and columns. And then also notice that the two methods agree for the yellow plots, right? So we expected this performance. If anything, we thought maybe the deep learning approach would perform worse, but we're finding that they perform essentially the same. So now let's start feeding both of our methods data that were not generated by the model we're assuming for likelihood. We're introducing model misspecification. We know this is going to mess up the Bayesian approach because this is a standard type of simulation experiment. We don't know how it's going to cause problems for the deep learning approach because it's an understudied problem at this moment. Okay. One thing to note, I forgot I had this slide, but one thing to note is that there are very real time trade-offs in terms of inference time. If we look at the time per inference for neural networks, we're talking milliseconds compared to hours for a Bayesian analysis. However, in order to get these millisecond long inferences for neural networks, I have to pay a flat cost to simulate my training data set. This is sometimes called amortization for the inference. How many inferences would I need to do for it to pay off? Well, if I need to analyze, let's say, 520 Bayesian models, my neural network suddenly becomes cheaper per cost, per analysis, right? So if you can design the perfect neural network that is applied 1 million times, you're going to be saving researchers, I'll make up a number, let's say hundreds of thousands of compute hours eventually, right? But that's a very large if with a very large asterisk next to it, okay? So let's get to our model misspecification experiment. I need to hurry along because I think I'm running low on time. So here are our experiments. Let's misspecify the basic reproduction number. In this case, our models both get the answers wrong, but look, they agree in how they are wrong. They both are wrong in the same way when you look at the yellow lines. Let's misspecify how densely we sample different locations. Both models are wrong, but look, the methods agree more often than not. They both fall on the gold points fall on the line. Let's misspecify how infections move between locations. Both methods have wrong answers, but they tend to agree with each other. Let's misspecify how many locations the model knows about. So we're going to simulate under a larger number of locations and then collapse them down to fewer locations. Both methods have the same amount of inaccuracy, but they tend to agree with each other more often than not. Let's misspecify what the input tree is because maybe we have finite sequence data and we don't have a good point estimate for the tree. Again, the methods, they both have error in their answers, but they tend to agree with each other more often than not. So over and over under different realistic model misspecifications, we tend to get similar answers between Bayesian and deep learning-based approaches. We then applied this to an extremely well-done analysis for the spread of COVID throughout European nations, published in 2021 by Nadeau et al. They have a very complex model. We're using a relatively simple model by comparison. But nonetheless, even though we used a relatively simple model, we ended up with per location basic reproductive numbers that are very similar to what they obtained. They're not crazy, right? And so if you keep this in mind, this is as close to a real empirical setting as you could hope to get because it is a real empirical analysis. We're finding that in simulation, when we misspecify models that mimic real empirical conditions, likelihood and neural networks perform similarly for parameter estimation. So is the case when we do this for a real empirical data set where we have no idea what the true data generating model is, okay? I'm positive we could close the gap between these two if we spent more time training models and so forth, but that wasn't the main point of the study. So I've shown so far that, in my opinion, neural networks under certain scenarios can accurately estimate parameters to equal levels of performance that likelihood-based approaches obtain. In general, when you misspecify models, neural networks and Bayesian methods are more similar to each other than they are to the truth, which is really an important result in my opinion, because it means that suppose you have a suppose you had a model and you could not get the likelihood function for it, but you wanted to conjecture how would it perform under a full-blown Bayesian analysis. These results could be interpreted as saying if you had that likelihood function, the neural network would be performing just as well as the likelihood function for those cases. So it suggests that potentially a neural network-based approach could give you likelihood-like performance even for models where you don't have likelihoods. That would be wonderful because then you can start thinking about models that are otherwise out of reach, right? Now, we've been starting to think about how you can apply these models to more complex epidemiological scenarios, such as the spread of viruses among primate populations and species, humans included, and in systems with vector-borne infections using ticks with an invasive species as ticks as a model system. But there's still this underlying issue that I'm interested in, which is there remains a fundamental level of distrust in terms of what can deep learning methods do, how under what conditions can we trust them, and how do we quantify the level of trustworthiness across a wide range of settings. And because of that, me and Ammon, we developed a new pipeline called Fiddle, which is designed to let people rapidly prototype simulation-based deep learning analyses to explore models that are otherwise not accessible through traditional likelihood-based approaches. And so the main goals of this are sort of the standard ones you'd expect for inference software. We want it to be relatively fast, flexible, and easy to use. It's meant to be reproducible, so we're using pipeline steps to manage workflow. And we want it to be customizable in the sense that users can set up simulators and design models in whatever way they're most comfortable. So we're trying to make it basically have a big tent that lets any type of biologist help explore phylogenetic model design. So the pipeline, a lot of this is built on our experience developing that first experiment I showed. And the pipeline has 5 steps going from simulation to plot. And I know I'm over on time basically, so I'll skip ahead. It has a simple command line interface. You can design a config file and write a simulator in any language to interface with the pipeline. We've been able to get this to work smoothly with macroevolutionary problems for parameter estimation, continuous trait evolution models for model selection, and we're even able to get, we were able to get our pipeline to produce accurate estimates that outperform likelihood-based approaches for outbreaks in that nasty phase of spread when we no longer have tractable likelihood functions. We're able to use these methods to obtain estimates that are very difficult to get under likelihood-based approaches, such as forecasts of prevalences. And it performs very well, at least under simulated conditions. We have a website with documentation. And I am going to... I'm just going to stop here because I think this is probably the most interesting one for discussion. Like I said, I come from a likelihood-based background, and there's a great amount of skepticism that I don't think is misplaced in what does deep learning or machine learning have to offer that we can't get from likelihood-based approaches. And I've had, over my time, many conversations with different people in phylogenetics who design models, and many people in phylogenetics who are what you might call applied biologists. And I'm paraphrasing, but this is a good representation of different attitudes I've encountered towards the role of deep learning for model-based inference, such as, that sounds interesting. I have no idea how it works, and I don't have time to learn about it. Like I have been and still am to some degree. I don't completely trust it, so I mostly stick to likelihood approaches when they're available, even if it means I have to sacrifice realism for the model. Um, some people are over enthusiastic. They say, perfect, now I can solve all the world's problems. Let's move on. We don't need likelihood anymore. That seems excessive. Some people I think they think it's powerful, but they also think it's dangerous. And I've gotten that in reviewer comments where, it's wonderful that there's this powerful tool, but if it's not used if it's not used perfectly well, then it has an immense capacity to be misleading. And it's not always easy to tell how you're being misled with it. And related to that, the field as a whole, in my opinion, is still unprepared to really understand and assess the quality of these inferences. Um, so I think I'm going to stop there because I know time is a little short. I would just like to thank Guppy again for inviting me and everybody for listening to me rattle on, especially 5 minutes over time. I'd like to thank some of my collaborators, and otherwise I'll stop here and open it up for questions. Thanks.
Thank you, Dr. Landis. I think we have so we're virtual this time, so we can stay a little bit late as long as you have time, but as soon as you want to leave, feel free. We appreciate it.
Sure, yeah.
But I'll go on mute and allow you to field the questions, but we'll kick it off with Nathan.
Awesome. Can you hear me?
Yes, I can. Hi.
Awesome, awesome. So I have done a little bit of deep learning work in the field of evolutionary prediction. And what we found, and I traditionally do stuff with protein language models and like transformer-based architecture stuff. But what we found is like when we're trying to do like conservation prediction and stuff like that, because it's one of the classical feature prediction tasks for deep learning, um, we found that like in certain scenarios, deep learning models tend to outperform more traditional phylogenetic prediction in the case of like when you have like poor MSAs and scenarios like that. Did you see any like scenarios where like you think that deep learning could like possibly be better? And then at the same case, do you think there are certain scenarios that like you would trust Bayesian or at least the traditional sense of models over the deep learning counterparts? Does that make sense?
Yeah, it does. So there are lots of parts of that question. I think so on a personal level, if you ask would I trust it, I still don't trust. So suppose you have a deep learning method that's trained on the same model as the model you specify for likelihood-based approach.
Yep.
I personally still do not trust that a deep learning method will outperform a
Ah,
Bayesian
okay.
approach if they're identically set up. However, there are lots of very subtle things that can give indications of better performance for deep learning than likelihood approaches that are hard to catch. For example, if I'm simulating trees under a birth-death model, you have to apply, for example, sampling conditions to what types of trees do you retain, such as do I stop my birth-death model when I attain n taxor or after time t, or do I throw away trees that don't survive until the present, things like that. Those types of conditions on the model are actually part of the data generating model. And it can be very hard to translate everything over to the deep learning model.
Hmm.
Sometimes I see differences in performance because of extremely subtle ways that we think about models, and it's not always clear how they translate across these different methods. But in general, if I see better performance from deep learning than likelihood approaches, I still always suspect that there's some type of training error,
True, true, true.
that the deep learning did not perform as intended. And the reason that's really nasty is because um, when you have other approaches, like say maximum likelihood, if you fail to find the global optimum, right? Because
Yeah,
you haven't run enough independent MLEs,
yeah,
you get a bad answer.
yeah, yeah, yeah.
If you run deep learning and it's not correct, you can get a better answer than you were hoping for. That's scary because you can be positively misled towards the wrong answer.
Yeah. No,
So
you
I still think it's best to always approach this stuff as, deep learning is probably not better than likelihood, but I could be wrong, right?
true, true, true. Okay. No, that's a great answer. Thank you so much for your
No,
time.
thanks. That's a good question. Um,
I think Omid was next, and then we have one in chat as
oh
well.
yeah, yeah.
Hey, thanks for the fascinating talk.
Hi, thank you.
Uh, I I was slightly confused by a part when you switched between testing on SIR and phylogeny, because I believe the salient feature of SIR models is capturing the dynamics, but the phylogeny works on maximum likelihood estimations and Bayesian stuff. I'm not convinced that a deep learning model which works well on one of them would necessarily work well on the other one.
Hmm.
Was it intended in your, was it studied in your comparisons?
Yeah, so I'd say there's two parts to that. The first part is what's the relationship between SIR models and say birth death models? And when you are in a under an SIR model, if you think about viral replication among hosts as a branching process, then during the early parts of an outbreak, only the early parts of an outbreak, that branching process can be framed as a birth death model. So they're mathematically equivalent. In the simulation experiments I ran, I was only looking at that early outbreak phase. So during that period, they are mathematically equivalent models. In the later phase of the outbreak, during that phase, we can't use a birth death model to approximate SIR dynamics, which I agree is extremely important. However, we can simulate SIR dynamics and see what rate parameters generate what types of prevalence curves over time and what phylogenies are consistent with those prevalence dynamics. And in those cases, we find that deep learning approaches do accurately reconstruct and even predict prevalence curves. Now, it's difficult because we don't have a likelihood-based method to do that at the moment, right? There are alternative methods such as piecewise constant birth death models and things like that, but we don't have a simple tractable likelihood function that can get at that dynamic. But in terms of whether it's appropriate to use phylogenetic models, you can basically view the prevalence as a the prevalence curve is sort of like the prevalence you would get for any random distribution of underlying trees. We just don't know what the tree is unless you provide the tree as information for input.
And
Mm.
I was some part wasn't clear too. Uh,
Mm.
did you use the deep learning model to get estimates for, did you say transmission rates or evolutionary rates?
This analysis was assuming we have phylogenies with
Mhm.
perfectly known topology and branch lengths. Okay.
Right.
And we know where individuals were sampled and we know when they were sampled. And from that, we are estimating the equivalent of an infection rate, the equivalent of a recovery rate, the equivalent of a sampling rate, and the equivalent of a migration rate.
Okay, so you're not getting estimates for parameters that are needed to construct the phylogeny.
No, we're assuming we have a phylogeny. Yeah.
Mhm.
There are other methods that are more focused on the tree inference problem, which is a difficult problem. We're just focusing on having a tree. Mhm.
Thank you.
No, thank you very much. Yeah.
Um, I will go ahead and read out the question from the chat from Dr. Casey Bergman. So you've shown that deep learning inference can work in a case where a machine learning model can be specified and therefore compared. The implication is that deep learning models can therefore work as well as machine learning models in other cases, but from what you say, this hasn't yet been shown. What
Mhm.
evidence would be needed in order to make the leap that, oh, sorry, maximum likelihood.
Yeah. I get that.
Um, to make the leap that deep learning models will work in cases where maximum likelihood models cannot be specified.
Yeah, it's difficult because it's sort like there is theory that any neural network can express any possible imaginable function. So if you have a likelihood function, in principle, a deep learning approach can approximate that function with sufficient accuracy. So that's one way you could approach the question. Another way is maybe by, you can't exhaustively prove that every imaginable model uh, could be equally well could be equally well fitted using deep learning versus maximum likelihood. because on one hand, we can't evaluate every model. Because there's infinitely many. But on the other hand, even for the models we could enumerate, many of those models won't have likelihood functions. So I agree there's a fundamental problem there in terms of how do you show the comparison when only one method is capable of the comparison. Now, um, you can still, within a model, you can still measure things like levels of accuracy, such as by simulating test data sets or validation data sets that aren't used during inference and measure accuracy that way. Um, and in principle, if you simulated large enough data sets that have infinite numbers of data points, you'd start to get these asymptotic guarantees you have under maximum likelihood as well. But those aren't satisfying from the theoretical perspective, right? That's an experiment showing that you have a relationship. But I generally agree. There is a little bit of there is an implication that is tempting, but I'm not sure how heavily we can actually rely on that holding true. That's a fair answer. Yeah, I hope it's true, but who knows? I think you probably get into again, I mostly come from a Bayesian background, so I certainly expect that deep learning and maximum likelihood share certain pathologies. Like if you have a non-identifiable model, they're both going to suffer in similar ways in terms of parameter accuracy. But that might be seen as a good or a bad thing. It's really mimicking it's then mimicking the good and bad properties of a model, right? Yeah. Yeah, interesting to think about. Thank you.
And then I think Luke might be our last question.
Hi.
Hey, hey, Dr. Landis. I really enjoy this talk because my work is very closely related.
Oh, thank you.
Actually, the sequencing effort of SARS-CoV-2 really makes the cold and standard like beast inference so computational challenging. My question for you first is about can you expand more on your simulation process, like how to get the phylogeny from this parameter, like what kind of tool you use?
Okay. I apologize that I didn't have time to talk about the software that I've been developing, Fiddle. That software is meant to essentially be a plug-and-play simulator, which means that if you have a method that can generate data sets and format them into a particular, like let's say CSV files and Nuix strings, if you can generate outputs that look like that, then Fiddle can fit those simulators. So we've been using RevBayes for simulating data sets. We've been using R for simulating data sets. We have a Python package called Phylojunction that we've been using to simulate data sets. We've been using the Beast plugin Master, which is used that does you know reaction diffusion equations for SIR dynamics. We've been using basically any simulator that seems relevant to phylogenetics and plugging it into the system. But the main issue is you need to have a well-defined mathematical model. You need to have a method that's capable of generating realizations under that model. And then you need to be able to do the window dressing that formats it into the expected format that Fiddle wants to read in. But what we've been working on, I think, is trying to address what you're talking about. Like, what type of simulator do I need to do these types
Yeah.
of analyses? We are trying to make it as generalizable on the front end so that if you don't want to spend 2 or 3 years futzing around with deep learning methodology, we can give you a relatively safe and well-behaved environment for you to do those analyses and at least explore the potential performance.
That's really cool and very helpful for us. The second question is like when you study virus dispersal, bees has this continuous model, like can modeling how the pathogen transfer across scale and time. I just wonder, like, is there like efforts like machine learning can do in solving this kind of like solving with continuous trait model, like coordinator kind of stuff?
Yeah, so although the main part of the talk only explored how deep learning performs for SIR models,
Yeah.
we have several examples for this fiddle package that include continuous trait models and model selection and things like that. So in principle, you could if you have a simulator, you could simulate the spread of viruses in two-dimensional continuous space under an SIR model, generate trees, and try to reconstruct, say, diffusion rates. Or, if you like jump diffusion models, jump rates between locations, things like that.
Mm.
But I think for that problem in particular, I don't know how many simulators exist currently for an analysis as complex as that. So, but one thing I think that's interesting about this entire problem is you sort of shift the bottleneck from can you get a likelihood function to can you write a simulator?
Mm.
And
Yeah.
that's a much lower burden because typically if you're validating a likelihood function, you have to have a simulator anyways. So you sort of removed one of the most onerous parts of method development, which is getting a likelihood function.
Yeah.
Yeah.
Totally great. Thank you so much. I really enjoyed the
No.
talk.
Yeah, thank you. I appreciate it.
So I want to be respectful of your time as much as I can. So if everybody could either unmute or I mean not unmute, but unvideo and add a clap reaction. Thank you again, Dr. Landis, for coming to share your recent research. We really appreciate it.
No, thank you. I appreciate it.
Thanks so much.
Thank you.